So in this section, we're going to look at the mechanisms of speciation. We've got speciation that occurs in separate places. This is separated by geography. That's allelopathic and sympathopathic. And this is speciation that occurs in the same place. So in allelopathic speciation, geographic isolation is what leads to the speciation. So when a population is divided by a geological barrier, and this geological barrier might be something large like the Grand Canyon, it might be a river could separate two populations so that they can't get over the river to breed with one another. It could be as simple as a lake drying up into different ponds, and so you have fish in one pond that are geographically isolated from a fish in another pond. The gene pools are isolated, and then you can have a change in allele frequency caused by natural selection, mutation, and genetic drift, and that can lead to speciation. So we've got pictured here two different antelope squirrels. That's what these are. And they live only a few kilometers apart, but each population is its own species. Because they're separated by the Grand Canyon, you can see these small little creatures couldn't get all the way down and across that river. And so they became, over many, many years, different species. So reproductive barriers evolve as the population diverges. So if you have geographic isolation, this creates opportunity for speciation, but doesn't necessarily lead to the new species. For the new species to appear, you need to have different food sources. Maybe if you're a flower, different pollinators or different predators. What you need is the population to be divided and to have different selective pressures, or perhaps even different genetic drift or mutations to occur. So let's look at speciation in fruit flies. This is a diagram of an experiment that was done by Diane Dodd. She raised fruit flies on starch or on maltose for 40 generations. That sounds like a long time, but a fruit fly can be born and produced young in about 10 days. So it took her, a little over a year to run this experiment. Fruit flies are chosen for a lot of genetic experiments because of their short generation time and because you can grow a whole lot of them it doesn't cost very much. So you can keep big populations so you don't get a lot of genetic drift. So she raised some of her fruit flies on starch medium for 40 generations, and some of her fruit flies on maltose for 40 generations. Now, after she had done that and raised a new population on starch and on maltose, she found that the starch fruit flies, could digest starch more efficiently. The fruit flies raised on maltose were able to digest maltose better. So she had actually changed their genetics a bit. And then she ran some mating experiments. And first we're going to look at this diagram over here. We have females, starch and maltose, males, maltose and starch. So it appears that there's a bigger number here for the starch breeding with starch and with the maltose breeding with maltose. There are small numbers of maltose and starch matches. But there's a predominance of the maltose individuals choosing to mate with maltose individuals and starch individuals choosing to mate with starch individuals. Now, this is our control, always want to do a control. Perhaps maybe it wasn't the starch and the maltose, maybe it was just that they had been raised together for 40 generations. So she took 2 populations that had been raised in starch. So both population 1 and population 2 are starch control groups. And we find an average number of matings. It doesn't really vary that much. So reproductive isolation is beginning by sexual selection in this case, because the starch individuals chose starch individuals to mate with, and the maltose individuals were choosing maltose to mate with. And so if the experiment had continued, we might expect that these intermediates here would become less and less. Let's look at another experiment that was done. This is a monkey flower, and generally the bees prefer pink. They prefer this pink flower right here, and the hummingbirds like the red ones. So the experimenters thought they would mess with their minds of the bees and the hummingbirds, and they produced the flower that the bees like to pollinate, but they changed the color. This has a red cast to it. It's not really a red, but it does have the red color allele. And when they changed it, they found that now hummingbirds would pollinate the pink flower or the red flower, and bees would continue to frequent it. If they change this red flower into more of a pink tone, then we start getting bees to visit it. Okay, so that's allopathic. Sympathetic speciation takes place without geographic isolation, and it occurs when species arrive within the geographic area as the parent species. So many plant species evolved by polyploidy. And this is the duplication of the chromosome number due to an error in cell division. So we start with the parent species. It has 6 chromosomes. Through some error or mistake, we get this cell here, and it has 12 chromosomes. When it makes gametes, the gametes have half the cell number, and so the gametes have 6. If they self-fertilize, we get another individual with 12. So we get a tetraploid plant. So this is a speciation that can occur in a single generation. Sexual selection can also lead to speciation. Here we have 2 kinds of fish, and they live in Lake Victoria. We have this fish, and I'm not even going to try and say that name, but they have red fins. The females prefer to mate with red-finned males, and the females see red better. They're more sensitive to red. Because of the wavelength of light is longer, red fins are visible in deep water. The other fish here has blue fins, and they prefer to mate with blue-finned males if they're female. The females are able to see blue better, and the blue light does not penetrate deep water, and so the blue is harder to see there, so they're going to breed in shallow water. And this will result in further divergence and further speciation.